back to Odylia Therapeutics Bridge Solutions video series. My name is Catherine Postamat, and today I want to talk to you about research tools. And as a patient advocacy organization, how do you decide if and when to invest in them? Research tools are critical for drug development for rare disease. They're really useful for understanding the biology of the disease, the protein, the gene of interest, but they're also really important for identifying potential therapeutics. Sometimes, depending on the disease of interest, they might already exist, but sometimes if a disease is newly recognized or if a gene and protein haven't been studied well in the literature, a real concerted effort might need to be made to generate these for the research community. And a patient advocacy organization can really be involved in order to improve timelines for the generation of these things, as well as to ensure that the research community has access to them when they need them, so that doesn't become a bottleneck later down the line. I like to start by talking about animal and cell models and how they can be so useful for understanding the biology of disease, the biology of a protein or gene of interest, the pathways involved, the different things that might be going wrong with different kinds of diseases. And they're really important for identifying potential therapeutics. These models are developed and they can be best utilized by understanding the specific validity that they have to a disease. When I say validity, validity is based on certain criteria, and different models can have predictive, phase, and construct validity. Predictive validity is a measure of how well a model can be used to predict currently unknown aspects of human disease. Another way of thinking about that is that predictive validity is the correlation between the animal and the human as it relates to therapeutic outcomes. So as we're thinking about this, an example might be a mouse model of a genetic seizure disorder. And if we treat that model with the seizure medication that we know works in humans, but it doesn't work in the model, then that means that that model does not have good predictive validity. Phase validity is a measure of how well a model replicates the disease symptoms in humans. So does the model exhibit all the symptoms and behaviors that are found in the human condition? An example of phase validity is a model of a disease which has many symptoms. So the question to ask there is, does the model exhibit the same symptoms as the human? If the model only replicates some or has different symptoms than in the human, then we would say it has low phase validity. Construct validity is a measure of how well the mechanism of disease in humans is matched in the animal model. So does the underlying biology match between the human and the model? So an example of this would be a model of a disease that causes narcolepsy. Is that condition in the model caused by the same thing that causes it in humans? If not, then the model has poor construct validity. While it would be ideal to have a model that has predictive, phase, and construct validity, this is rarely possible. But important findings can be made in models that don't perfectly replicate the human condition. However, it's necessary to understand the limitations of a particular model being used in order to interpret results correctly. And different animal models are good to answer different questions, and some are better suited for different diseases. And that can be based on the type of genetic mutations that are involved. So for example, some diseases cause a reduction in gene expression, a reduction in protein expression, while others have an increase in both gene and protein expression. So that can really dictate sort of what kind of model makes sense for that specific disease. And examples of models that exist are mice, zebrafish, worms, cats, dogs, pigs, primates. There are many. A lot of the larger model systems are naturally occurring, so we might find in a larger type of animal that it gets a disease that is similar to humans, and later we find out, oh, it's also caused by the same gene, and that can happen naturally. But it's really important to know that each model has benefits and weaknesses, and it's really best to ask questions to be sure that the model system that's being used is best for your specific disease of interest and the questions that you're asking. And so the best thing to do in that case is talk to researchers in the field and researchers who are studying your specific disease and ask the questions of, is this the right model? And are there better ones out there or additional ones that we could add on to? Another really important research tool are human cell lines. And human cell lines can be created either through gene editing, through techniques such as CRISPR, or from collecting cells from patients. One type of cells that we often collect are called fibroblasts. Fibroblasts are a specific type of connective tissue that's found in our skin and our tendons. It's really easy to collect and it's pretty easy to grow in the lab. And it's a great way to study the genetic background, the specific gene of interest in cells directly from patients in a way that patients aren't needing to repeatedly come back again and again and again and give samples. And fibroblasts are also what we call reprogrammable. And what we mean when we say reprogrammable is that scientists in the lab can take that fibroblast skin cell and turn it into a neuron or a kidney cell or a liver cell or another type of cell that is specific to that disease. And in that way can look at how potential therapeutics can affect specific cells from patients. So building off of fibroblasts are another type of cell called iPSC cells, or induced pluripotent stem cells. And these type of cells are derived from patient-donated skin or blood cells that then can be reprogrammed into first stem cells and then made into any number of different type of cells. And this process can take months, up to 6 months in some cases, but allows for the generation of specific targeted cell types, again with the genetic background of specific patients that have the disease. And in this way, researchers can look in a dish and learn about early cellular, microscopic, really small changes that are happening in different pathways and cellular processes that can lead to disease. And in that way, learning about the biology can help identify potential treatable, druggable pathways and allow for testing of different therapeutics before moving to a multicellular or a larger model organism in order to test those therapies in. So the last research tool I want to talk about today are antibodies. And we often think about antibodies in terms of our immune system and how they help us fight infection, but they're also a very useful research tool. And that's because antibodies are really good at recognizing specific proteins and binding to them. And because of this characteristic, scientists and researchers have been able to use this ability of antibodies to bind proteins in order to visualize proteins in cells and in organ systems. And that can help us understand where a protein is in a cell or in an organ and how much of it exists. So it's very important to have a reliable antibody early on in studies, so we can sort of throughout the course of a study determine an effect on protein and where that protein is in a cell, if it should be in the right spot or not, as well as really critical in late stages of therapy development, because having a working antibody allows us to prove that a particular therapy is having an effect on that specific protein. So what makes a good antibody? A good antibody has a number of characteristics, including that it's specific, meaning that it's specific to the protein of interest. It also provides clean, easily decipherable results. A good antibody is broadly accessible to the research community, meaning oftentimes that it's commercially available. And it has been proven to work in different labs. Sometimes an antibody works well in one lab, but not in another. But a good antibody is able to work in multiple different labs. A great antibody is an antibody that can be used in multiple different types of experiments. And why this is important is because an antibody can tell us not only how much protein is there, but also where a protein is. And this is really important because proteins generally have specific functions that occur in either specific cells or in specific regions of the cell. And merely increasing the amount of a protein might have no effect or even a negative effect if the protein's not getting to where it needs to be. I hope you enjoyed our video on research tools. Please feel free to like and subscribe to this video so you can get access to all of our videos as soon as they become available. And please feel free to comment and ask questions down below. We look forward to seeing you in our next video.